So you said for meiosis, we do not need to remember because it's the same for mitosis. The steps of the cycle: metaphase, anaphase, telophase, and cytokinesis. We don't have to go back over that.
You don't have to know what goes on,
Okay.
just
Thank you.
points to know you remember, so.
Okay.
Okay, folks, today, we're going to look at how you set up these squares. And, there is a way you can do this without having to do the squares, quite honestly. Mathematical way, figuring out ratios. If you take some biology, then you'll figure out how to do it without having to do these. I want to, getting the things set up is the most important thing. So once you get them set up correctly, the rest of it is quite easy. Most people on these, if they mess up, it will be because they did not set it up correctly. And usually on this exam, these are worth about 20 points. So they're worth a lot of the test. Now, if you remember last time, we talked about the fact that for single traits, if we start with pure parents here, pure parents there, recessive, those are homozygous dominant, homozygous recessive. Each parent can only contribute one allele. This one can only contribute a dominant trait, that one can only contribute a recessive trait. Because of that, when two pure parents are born, all of the offspring will be heterozygotes. Heterozygotes mean they've got one dominant trait and one recessive trait. So all of the offspring have to be that, and that is basically just a 100% ratio there. Now, if we take two of these and make them, remember, this is not a have-to case. The problems will still work out if you don't do it this way, but you should put the female here and the male at the top. That's just the way that it's done. It's just a consistent way. It's the way everybody else does it, so people will know what you're talking about, and you should know what they're talking about. Notice when we take these particular individuals, since this is what genes they have, what genes can they give to offspring? This can give either the dominant trait or the recessive. Now, which one, what is the probability that they will give those? So we come here, we put the female there, the male. In this case, that's not the same one. Here, they're using a homozygous recessive, and then they're using one of the heterozygous, so they are not done F1 generation. If this had been the F1, we would have had a dominant trait there. So they have taken, this is not the F1, this is F1. And this is taking one of the F1s and then another flower that has this genotype crossing it. But the key is the same. Once you get these set up, you just, in those squares, this P goes to that square, that P goes to that square. This P goes to that square, that P goes to that square, this P goes to this square, this P goes to this square, this P here, that one there. That's all you do. The key is getting is set up, the genotype. Remember, the genotype is going to be the actual alleles, how we write these. That's the genotype. Phenotype is what do they look like, purple and white. Those are pretty easy when we're doing it this way. Now this is where we take the two pure parents and cross to the F1 generation. We always get that 3-to-1 ratio. Don't forget that ratio, because you will see it and hear it again, and when you do this in cell biology, when you're studying genetics, and you will be expected to know that the product of two pure breeding parents will be heterozygotes in the F1 generation. And in the F2 generation, if you cross to the F1, you always get that 3-to-1 ratio. Same process. This one goes here, that one goes there, that one there, there, this one here, that one here, this one here, that one here. So setup is key. There are sometimes traits, sometimes they're linked to each other, and sometimes they just relate to each other, like for color or things. They're not necessarily tied to each other. They separate independently. Which alleles you get are independent. It's just a shot, it's random, okay? That's what we're going to look at, and we are going to do 2 of these. Now, when we do 2 of these, this is for flower color. So you've got the 2 alleles for flower color. Now there will be 2 more alleles over here, and that is not going to be just for color, but it's going to be for the shape of the seeds. Are they round or are they wrinkled? And once you learn how to set these up, it's a breeze. So, here we go. Now, walking through this, we have 2 pure breeding parents. This parent is homozygous dominant. Homozygous dominant both for color, which is yellow, and for seed shape, which is wrinkled. Homozygous, or here it's the seed shape is smooth, and the recessive is green and wrinkled. Okay. Now, how do we come up with these letters right here? And we do it exactly the same way. What can this guy give right here? He can only give a dominant trait, a dominant yellow, and can only give a dominant round. That's all he can give. Same over here, except this one can only give the recessive green and recessive wrinkle. So how do we combine? What are the possible matches we could come up with? We could come up with a large Y and a small y. We can come up with large R's and small R's and so forth. So how do we combine these? The gametes, the offspring, the F1 generation, remember, will always be heterozygotes out of pure parents. So these will be the only 2 gametes that this can give alleles, the only 2 that one can give. So when we put them together, we've got a heterozygote. Now, do you follow up to that point? All this one can give is the dominant color, dominant shape. This one can only give the recessive color and recessive. To all the choices. Do you understand that? Make sure. So that's what their offspring looks like. This is the F1 generation, remember, that will always be 100% heterozygous. Now, if we cross two of these, what do we come up with? There is a very simple way of doing it. It's almost like in math where you have to multiply everything in the equation. If we take one of these and multiply it by that trait, that's one possibility, we put it right there. We can take that large Y and then go with the small r, put it right there. Then we have to do it with this other Y. Multiply that times the big R, and then multiply it by the small r. That's all there is to it. But make sure you understand what just happened. We've got, this is the genotype, so we take this one, combine it with that one, this one, combine it with that one, then we take this one, combine it, this one, combine it. And we get this, and it will be the same on top because we're dealing with two of the same generation, F1 generation. Now, is there any question on that setup right there?
If we had, so it's not always going to be homozygous dominant for yellow and color recessive. Okay. So
You will always have to be given the genotype.
right.
You have to know what the parents are. You can't figure that out. You can figure it out if you've got a bunch of other information. But when you're doing problems on tests in most places, you already know what the two genotypes are.
Okay.
Now, how do we combine these the same way? This and that, this and that, right there. Then we take this and that, this and that. Then we take this, and that one, and this, and that one. See how we got everything? Now they're combined. Now here is something also that can save you a lot of work. It is going to be the same for all of these. Okay? Notice, when you do these and fill these all out, what you are going to get, when you combine these, it is the same for all of them. You do the same thing. Take this one, Y, and big Y right there, R and a small, or a big R right there, and just keep doing it. Remember, this one goes in these boxes, then this one goes in these boxes. Now, when you do that, and you get all of these combinations, you go in and count how many round yellow seeds were there. So we count 1, 2, 3, 4, 5, 6, 7, 8, 9. How many round green seeds will there be? There is going to be 3. How many yellow wrinkled seeds? 3. And how many green wrinkled seeds? 1. This is another ratio that will never change. When you multiply, or when you combine, mate, 2 heterozygotes from the F1 generation, you will always get this ratio. It will never change. 9, 3, 3, 1. Just as the heterozygotes for one trait, a monohybrid trait, is always 3 to 1. This is always 9, 3, 3 to 1. Any questions on that? Once again, the key is to set this up correctly. And then And I will ask you questions such as, how many brown green seeds were there? How many wrinkled green seeds were there? You've got the square there to tell you. And you can do this down at the bottom. This is your ratio. Notice there's 16 squares. 9 out of the 16 were the round and yellow. 3 out of the 16 were round and green. 3 out of the 16 were wrinkled and yellow. And 1 out of the 16 was wrinkled and green. That is your ratio. 9, 3, 3, 1. And you can get into some wild combinations with this. We are talking about only one thing, complete dominance. There's other things that happen. That can, if you've ever seen flowers that'll have like white leaves and all these purple splotches or red or whatever different colors spattered throughout, that's another kind of inheritance of dominance. It's not complete dominance. You can get bits and pieces.
I was wondering, so on the test, you will want us to provide the phenotypic ratio and the,
You will have to do one of these
okay,
on it. You'll have to show it. You'll have to do one. You'll be given the genotype, or you'll be able to figure it out.
okay.
And usually, the questions will be something like, if we have a white heterozygotic mouse, what does that tell you?
That white is dominant.
That white's dominant. It's heterozygote, so, and it's white. Now, if we say a white heterozygotic mouse is crossed with a brown mouse, what's the genotype of the brown mouse? It has to be homozygous recessive for it to be that recessive trait to show up. Right? Now, that's the kind of question you're gonna get. You're gonna have to think and figure this out. A heterozygous white mouse with wrinkled hair is crossed with a brown mouse with straight hair. Which is dominant, wrinkled or straight? Huh? Wrinkled. It's heterozygotes, white mouse with wrinkled hair. So it has to be white's dominant and wrinkled hair is dominant. The brown is recessive and straight hair is recessive. Now you can get the information if you look at them that way.
How do you decide which one's dominant, the white or the brown?
What shows up?
I mean, you don't just like pick one, or?
No, you'll be told a heterozygous white mouse. If it's heterozygous, the dominant trait shows up, right? So it's white. And the way that you figure out what trait's dominant is what shows up. And in nature, one trait might be dominant with this combination, but be recessive to something else.
So I mean, you just pick which trait you want to be dominant, though, like, I get the mouse one?
No, you don't pick. I'm telling you, and the question will say,
Right.
a heterozygous white mouse.
So whichever animal you start with.
Huh?
Whichever animal you start with. Like you said, it's white mouse and a brown mouse.
I will tell you though, are they heterozygote or not?
I get it, yeah.
So if it's heterozygote and it's white, it has to be dominant. And I will pick that and put it in there. You won't have to choose anything like that. You will just take the information given and decipher it. Heterozygote and it's white must be dominant. Heterozygote with wrinkled hair must be dominant.
Gotcha.
So that's the way you figure them out.
Are you gonna make sure that what?
I'm going to tell you to do a dihybrid cross. And you will have to do all of that, and then there will be questions. How many brown mouse with wrinkled hair? How many white mouse with straight hair? Things like that. And you would count it. It will always be in that ratio of 9, 3, 3 to 1. That will never change. Unless, what if I give you a heterozygote white mouse is crossed with a mouse that's brown. So you no, because white's dominant, brown has to be recessive. But is heterozygotic for straight hair or wrinkled hair? See, what I'm trying to say is, don't expect to get two heterozygote mother and father. They're gonna be big. You could have one that is dominant for, let's look at these. Notice this right here is dominant for the color, but it's homozygous recessive for the shape. You can have different combinations. Now, I could have picked this one right here and crossed it with that one. Ratio's now going to change. It won't be 9-3-3 to 1. It's 9-3-3 to 1 only when we have heterozygotes cross. So in this situation, I can pick any one of these, and I might just keep the seed color here for green, whatever. It doesn't matter what sample is. But I can say right here that we have got a heterozygote pea that is yellow and round. So what does that tell you? If it's heterozygote and it's yellow and round are the dominant traits. And then if I say there is a green round seed, you know those have to be homozygous recessive. And that one right there has to be the dominant trait.
So basically, with the mouse example, you can say that we have a white heterozygote mouse. So we know that white's dominant, and then you can say that we have a brown mouse, so we already know that brown would be recessive if white's dominant. And then with the texture of the hair, either straight or wrinkled.
Just whatever combination I want to put in there, it doesn't matter.
Right, okay.
And you can do it with flies, you can do it with plants, you can pick any color you want. I could have just as easily have said a brown mouse is dominant to white. It doesn't matter. The only way you know about that is what shows up. The only way you can tell.
Also about so on the right we have independent assortment, and on the left we have dependent assortment.
A hypothesis of it, which is not good.
Okay.
It's bad. Don't just forget that.
Okay.
That's just showing you an example of way some people might think, but it's wrong. I wish they wouldn't even put it in the book. Forget that. So, everybody good with that? You can set one of those up. I won't try to make it too confusing for you. You will have to do a simple one, a monohybrid, and you'll have to do a dihybrid. Don't get misled. And you can do this for genes in people, such as a gene that will produce a certain immunoglobulin. And if you take genetics, you'll figure out there's a way we can do this with just numbers and little math tables, and you don't even have to write all this out, fill it out, but you have to know how to do that in order to do the numbers. But there's a way of shortcutting. Always. Okay. So let's move along. Make sure now, like I say, these two squares, the mono and the dihybrid, usually are going to count for anywhere from 20, 25 points off the exam. So if you mess it up, it's not a good situation to be in. And I do give partial credit, like if you get part of it right, you mess up on something else, so I do cut you a little bit of slack there. So if there is no further questions, the only thing this law of independent assortment, that means one of these that you're going to get, you don't know. It's a total chance. It's like putting all of those peas into a bag and just picking one out. You don't know if you're gonna be an offspring, which all of these represent offspring. You don't know if you're gonna be this one or that one. And there's nothing that guides it. It's just telling you a chance. If you put 4 peas in a bag, one was the round and yellow, and one was the round and green, and one was the wrinkled and yellow, and one was the wrinkled and green. If you put those in there, you would have a 25% chance of getting any one of them, would, and it would be total chance. It wouldn't be anything guiding it. And if you put 16 of them in with this ratio, it would say you would have a 9 to 3 to 3 to 1 chance of getting these. Now you can turn those into percentages. You can divide 9 by 16 and come up with, you got that percent chance, right? So if I actually were to get in there and to do something like that, divide 9 by 16, I get a 56% chance. If we put 16 of those peas in this shape in a bag, 56% chance that I would draw out a yellow round pea. And the others would be an 18% chance that you would get a round green one, an 18% chance that you would get a wrinkled yellow, and then there would be a 6% chance that you would get a wrinkled green. So you can take those ratios, remember that is out of 16, and you can turn that into a decimal. The easiest way is just to do the ratio, unless you're asked directly what is the percentage chance, then that's the way you calculate your percent chance.
In that case, would we need to have our calculators for the exam?
I don't know if you would or not.
So
You
should
can
we?
use one for it, yeah,
Okay, thank you.
but it's not that difficult to divide the longhand. You're dealing with just small numbers here. 16 will go into 90 how many times? 5.
It's only once.
You can almost do it in your head. Now, law of independent assortment says it's all the chance, like if you just put all those possibilities in a bag and pick one. So there is nothing guiding it, is the key. These are 3 of the kinds of dominance. One of these causes a lot of problems and a lot of variation also. We just looked at the flowers and the color was either one way or another. One gene totally masked the others. That is called complete dominance. The phenotypes of the heterozygote and the dominant homozygote are identical. In other words, if you have the dominant allele show up at all, that is what is going to be present. You don't have to have both of them, you can just have one, and that is what shows. Incomplete dominance is a little bit different. The phenotype of the F1 hybrids is somewhere between the 2 values. So when if we had a red flower and a white flower, the offspring will be pink. For the heterozygotes, and we'll see that. So the heterozygote looks like the dominant one in complete dominance. In incomplete dominance, it looks like a blend. Now, it is colored heavier towards the dominant one, more reddish pink than whitish pink, but it still is blended. And you'll get to see that in a minute. Codominance is one that is very, very important. Codominance, both of the alleles show up in the phenotype. And the best example of that is when you look at roan cattle. You've ever seen roans? Roan cattle are mostly red, but there are white hairs growing out of it. Now, it's not a blended, it's a pure white hair. It's just mixed with a bunch of red. That's codominance. Where the dominant shape trait shows up more than the recessive, but the recessive still shows up. So when you see that in cattle, you get that kind of splotchy looking cattle effect. In codominance, there's some other things that this is really important in too. Here's something new for you to start thinking about these alleles. Remember, what does a gene code for?
A protein?
A protein. Now, how much protein can be affected by whether it is homozygous dominant, whether it's heterozygous, or homozygous recessive, whatever. In other words, it's more codominance here. The amount of a protein that is produced can have enormous effects on the way the body works. Most of the things that these proteins do, there has to be a certain amount of that protein produced in order for the function to be normal. If you don't have that much, you can have a disease. Okay? Think about insulin in pancreas. You have to have a certain amount of insulin in order to function normal. What happens if you don't have enough? Then you become a diabetic. Now you can still have some insulin there, but it's just not working the way it's supposed to. There's many things in medicine where the effects are going to be how much of the protein is produced, and that is going to be determined by what the genotype is. And in genetics, we'll talk about a lot of these diseases and things that pop up. So here we've got three ways that the phenotype can be expressed, basically. Complete dominance, whatever the dominant trait is, shows up. In incomplete dominance, there is a blend between the dominant and recessive trait, but the color, for example, will be more towards the dominant trait. It'll have more of that color in it than the recessive. In codominance, they both show up. Here is a case of what kind of dominance. Now, we've got, here is another way that you are also going to see some things written. Instead of just using R's and W's, there will be one letter that represents a particular gene characteristic. That large C right there is there just to represent color. Then they use a superscript to denote which color, red and white. The way we were doing it, if you go back, we just used the letters. We didn't have a superscript, it's just a P. Now, if we had done this the other way, there would have been a C, and then the P would, capital would have represented purple, and so forth. So, don't let that fool you right there. This C represents the gene. This represents the allele, the version of the gene. So we've got a dominant red, pure generation, and a white. notice it's also what?
Dominant.
It's not written as recessive, is it? So what do we get? We cross these two colors. We are going to get the heterozygote. What is the heterozygote? In this case, it's going to be one of each, right? Two different alleles. So, when we do our little Punnett square, we've got CR will go into one cavity, CW into another. That's the two choices that we've got. So here's CR and CW, and then CR and CW for the male. Cross them the same way. R is dominant, red. Homozygous, red. R and W is heterozygote for the two traits. CW and R is another heterozygote, and CW is now the homozygous white color. So look at the ratio that we got. What type of dominance now is this?
Incomplete dominance.
Incomplete. Remember, incomplete is where you will get a blend of the traits. The homozygous red will still be red. The homozygous white will still be white. It's just that when you look at these, when they are heterozygote, the color blends. It still blends towards the dominant trait, which in this case is red. But red and white, that's the way, then you come up with a pink flower. They blend. If this had been complete dominance, then we would have had, out of that F2 generation, 3 red and 1 white. Because these pink would have been enough to take that dominant red color. Relationship between dominance and phenotype. The dominant allele does not subdue the recessive alleles. It's not the way they react. Alleles are simply variations in a gene's nucleotide sequence, in the gene sequence. For any character, dominance, recessive relationships of alleles depend on what you see, what shows up. The phenotype, how does it look? Here's a disease that will give you an example how important some of these things are in the proteins that are going. At an organism level, the allele is recessive. So if you carry the allele for Tay-Sachs, you must have both in order to see the disease. If you only have one, then you would be a carrier for the disease. At the biochemical level, the phenotype, the enzyme activity level, is incomplete dominant. At the molecular level, the alleles are codominant. So it depends on what level that you're looking at. And Tay-Sachs is a disease. There is a covering, a protein covering over all of your nerves called myelin. That myelin speeds up transduction of nerve impulses, smooths everything out, and makes your nerves work fast and smooth. On occasion, this myelin, just everyday general things, starts to wear out, so the cells of your nervous system, a nerve cell has it's not a neuron. A neuron is in the brain, that's the nerve cells in the brain. The nerves that come off are called peripheral nerves, and they're a different structure. These nerves have the ability to do repair to an extent on that myelin. In other words, as that myelin degrades, cells can make more myelin and get rid of the old. Tay-Sachs is a disease where there is a problem of getting rid of the old myelin. And what gets rid of that old myelin is what does all the work in the body anyway, enzymes. There is an enzyme, a particular enzyme, that will destroy that myelin, get it out of your system. If that enzyme is non-functional or not there, the myelin builds up, and it builds up in the brain. And when it builds up in the brain, it then destroys brain tissue and eventually will kill the individual that has it. So it's a very, very important little enzyme. And it's all that whole disease, a fatal disease, is because your body lacks one simple enzyme to get rid of that waste. And what this is saying, if you look at that combination at the organism level, the individual, the allele is recessive, so you have to have it on both chromosomes. At the biochemical level, however, the phenotype is incomplete dominant biochemically. In other words, both of them are producing, but maybe not at enough of a particular level to do what needs to be done. And codominant, they're both working, but once again, it's not enough being produced to do the job. This right here, this says lipids. When we were talking about the myelin, you come to find out there are things associated with the myelin that makes it up called sphingomyelin, which is the myelin, but it's the fat attached to it, the protein molecule that builds up and causes some damage, in case anybody was wondering. Now, alleles or variations of a gene trait in the population. This is getting into more biology for organisms now, like you see out in the wild. It says that most of these genes will have more than two versions of an allele. In other words, we don't just have black hair color or brown hair color. You can have blonde and different shades of brown and red and so on. When we look at blood, and most of you, if you've been in the 1081 labs, you've done blood typing. Blood typing, first off, is not as straightforward as people may think. There are actually, I think, 14 different antigens on a red blood cell. Fortunately, there is only one that will produce a severe enough reaction to do us harm if they're not matched up correctly, and that is the AB antigens. So we devise blood grouping, we call it the ABO blood group. Now remember, if you have an A antigen, that means the antigen that's on that cell, when it comes in contact with another A cell, won't do anything because it's an antigen that is devised to accept A. If, however, it comes in contact with a B blood cell, with a B-type antigen, then the A will release from its cell antibodies to fight the B. These cells carry a supply of whatever antibody they carry inside the cell and can release it when they come in contact with another cell. So type A antigen will carry anti-B or antibodies, anti-B antibodies, and will release them when it comes in contact with it. B will carry type A and so forth, and you know, you've heard things such as the universal acceptors and universal donors. AB can take about anything. O can donate to anybody, but can only take O. Plus the positive and negative, or H factor plays a big role. But look at how this is set up. It says there are 4 phenotypes of the ABO blood group. You can have either A, B, or neither. The small case I just means that you don't have either entity. So your combination, you can have be A, you can be B, or you can be AB, or you can be O, and neither. The enzyme encoded by the A allele adds the A carbohydrate, whereas the enzyme encoded by the B allele adds B carbohydrate. The enzyme encoded by the I allele doesn't have either. These are the alleles and the blood type, and it's showing you how you can have different versions of these antigens. Notice here, that's the shape of the A, that's the shape for the B, and this is both, and this is one that doesn't have either. So, if you've got A blood, you've got 2 versions that you can have. You only got 2 choices if you got A. You can have one parent give you the type A, the other parent give you the type A, or one gave you neither. It gave you this I. So the only one factor there is an A, it will be A, that be your blood type. If the only factor you have is B, then it shows up as B. And right here, when you look at these now, remember, it can either be homozygous, or it can be here, you can have one allele that has a version and this one doesn't have anything, so since that's the only one there, it shows up. Here you have both. Now, and we ask this question, can a person who is type AB be the father of a child that's type A? Of course. He can give either allele. He can be the father of someone who's type A, father of someone who's type B, father of someone type AB. Can he be the father of someone type O? No.
Why did you say though that the AB could cause a dangerous reaction that comes into contact with other, that's like the worst one?
It's not necessarily the worst one, but O is one. It's probably the worst. O, while it doesn't have either antigen, it's got both anti-A and anti-B antibodies inside. So, and when that happens, if you give blood, they have to give it very slowly, very gently, because if you rupture those cells, then it releases the anti-A and anti-B, which they've already got in their body, and it will attack them. AB has both antigens and doesn't have any of the antibodies, so it's actually one of the safest. O is called universal donor because you can give it to anybody because it doesn't have it, but only O can take only O because it's got both antibodies inside.
Because I always hear that blood banks like that.
Most, well, if you look at the rhesus factor, the RH factor, guys, the RH factor, positive shows up in 85% of the people, negative 15%. So the most uncommon blood type out there is O negative. And that's what they need and want. O positive is still kind of rare because it's O, but not nearly as much as O negative.
Oh, sorry.
We'll pick up there next time, guys.
How do you determine which alleles the offspring will get? I mean, either the first one